Hello students. Today I discuss about ribosome, the very most important cell organelle. Ribosome. This term first given by Pallet. Okay. Then what is ribosome? Ribosome is a spherical non-membranous cell organelles which is present in eukaryotic and prokaryotic both cell. These are the ribosome. Ribosome is the cell organelle which is present both in eukaryotic and prokaryotic cell. Okay, that's the non-membranous cell organelle. Very important part. And the ribosome composed by RNA and protein. RNA is ribonucleic acid and protein, the composed ribosome. Okay. Then ribosome is the smallest cell organelles, and ribosome also called organelle with organelles because ribosome is the organelles and it is present in the organelles of plastid in mitochondria. That's why it is called organelles in organelles. And also ribosome called is the protein factory because it helps to synthesis of protein. Okay. Then origin of ribosome. Origin. The ribosome origin from nucleolus and with help of cytoplasm. Mainly it from nucleolus and with the help of cytoplasm jointly. This is the origin of ribosome. Then types of ribosome. Types of ribosome. One is cytoplasmic organ ribosome and organelle ribosome. Two types, cytoplasmic ribosome and organelle ribosome. Cytoplasmic ribosome, it is present in cytoplasm, this is free, and organelle ribosome is present in mitochondria and plastid. This is the organelle ribosome. Another types, it's 70S, the eukaryotic and prokaryotic. In case of eukaryotic, is the 80S ribosome. In case of prokaryotic, is the 70S ribosome. What is the meaning of S? S means sedimentation coefficient. When is centrifugal, during centrifugation, in what rate the ribosome is settled down, this is denoted by S. That's the sedimentation coefficient. Okay. Then we know the most of the eukaryotic and prokaryotic cell contain this ribosome. But in mammalian RBC and mature sperm does not contain any ribosome. Okay. Then eukaryotic ribosome is 80S and prokaryotic ribosome is 70S. But in eukaryotic cell also present 70S ribosome in mitochondria and plastid. Okay. In eukaryotic cell, 70S ribosome also contained in mitochondria and plastid. That's the introduction of ribosome. Then structure of ribosome. Structure, in case of prokaryotic, 70S ribosome, and it has two subunit. One larger subunit is 50S and smaller subunit is 30S. 50S when the larger subunit the settle down in rate of 50S and smaller subunit settle down the rate of 30S. Okay, this two subunit, and here present 60% are RNA and 40% protein. 40% protein is present in case of 70S ribosome. Okay, and rRNA present, which type of RNA? Present 23S, 5S and 16S. This 23S and 5S form the larger subunit 50S and 16S form the smaller subunit 30S. Present in the 16S present in 30S smaller in subunit. Okay, and in case of 80S means ribosome, 80S ribosome, the larger subunit is 60S and smaller is 40S, and they are present 40% RNA and 60% protein is present, and rRNA present 28S, 5.8S, 5S and 18S. These three present in larger subunit 60S and this is present in smaller subunit 40S. Okay, that's the structure of 80S and 70S ribosome. Then the two subunit, the structure in 70S and 80S almost same. First, larger subunit. The larger subunit, suppose this one the larger subunit. It has three parts: protuberance, ridge and stalk. Protuberance, ridge and stalk. These are the three parts present, and they are present a tunnel-like structure also. What is the function of this tunnel-like structure? Now this subunit form the polypeptide which transfer through the tunnel to the endoplasmic reticulum, and this ER helps to transfer this peptide during protein synthesis. Okay, and this larger subunit also have three sites. One is A site, another is B site and another is E site. A site means amino acyl site. A site means amino acyl site. Amino acyl site. P site means peptidal site. Peptidal site. And peptidal site. And E site means exit site. These are the three sites present. Okay, and in case of smaller subunit, they are present a groups. What is the function of group? It is the mRNA binding area. In this place, mRNA is attached during protein synthesis. Okay, it has also four parts: head, base, platform and cleft. Head, base, platform and cleft. These are the four parts is present, and most of the time these two units are separate. They are not joined. When they join? Now during protein synthesis they are joined to each other. Okay, during protein synthesis. And there also another thing is present, magnesium. This is the very important part, take part during protein synthesis. If the magnesium concentration 0.001 molar, then two subunits are attached and helps to form protein synthesis. But if the concentration is the less than the 0.001 molar, then this subunit is dissociated, occur dissociation. And if the concentration of magnesium ion more than 0.001 molar, then two ribosomes are attached and form dimer. Okay, so the concentration of the magnesium ion during protein synthesis is very important. Okay, and another thing, when the more ribosome present, this group of ribosome is called polysome. This is the very important. And the main function of ribosome is protein synthesis. This process translation I will discuss in next video. Okay, that's about the structure of ribosome. If you have any doubt, any queries, you can comment. Okay, student, thank you.